Good morning, judges. We're from Stella High School. Our project is called In-Vitro activity of plant extracts and commercial cough medications against S. aureus and E. coli. Commercial cough medications tested were strepsils, woods, coffee 100, while Chinese traditional medicine tested were Changpei, Yingqiao, and 24. Some examples of natural products are apple cider and honey, which are all tested for their antibacterial activities. Different commercial cough medications and herbal teas were tested for their antibacterial efficacy, and results were compared to the antibacterial results obtained from the natural product extracts. This research was conducted to produce a natural product remedy that is an effective alternative to antibiotics in treatment of cough caused by bacterial infections. Now my teammate Kelvin will talk more about the procedure.
1 ml of each Western medication and drink sample such as Coke, apple cider, and honey is extracted out and then placed into a sterile microtube. However, the active ingredient in each of the TCM and raw herbs is boiled in 25 ml of boiling water, and the solution is then placed under UV irradiation to eliminate any pre-existing fungal and bacterial infections. 1 ml of each solution is then extracted and placed in a sterile microtube. 1 ml of each sample is then mixed with 0.1 ml of each bacteria, once with S. aureus and once with E. coli, to obtain 1.1 ml of antibacterial assay test sample. The solution is then incubated in a water bath for 24 hours and then plated onto a LB agar plate. Colony forming units was determined after 4 and 24 hours of incubation. In this study, we also tested for the synergistic and antagonistic effect of mixtures of medications. 0.5 ml of the first sample was added to 0.5 ml of the second sample to obtain 1.0 ml of the sample mixture. 0.1 ml of each bacteria was then added to the sample mixture, and similar incubation and colony forming unit determination methods is used. Now I'll pass the time to Kenneth, who will talk more about the results.
Results have shown that Western medicine worked better than traditional Chinese medicine. However, from the chart, it is noticeable that traditional Chinese medicine worked best on the basis of 24 hours. Therefore, it can be concluded that traditional Chinese medicine worked best every 24 hours, having an average of 50% drop against S. aureus and E. coli. On the other hand, Western medicine showed close to immediate effects on the absorption of woods against S. aureus and strepsils against both S. aureus and E. coli, having almost 100% reduction in a short period of 4 hours. It can be seen that strepsils is the weakest among the three Western medicine. Therefore, we can conclude that strepsils is only suitable for reducing small concentration of microorganisms. Results have shown that Forsythia worked best amongst the raw herbs and home remedies tested, followed by lime and ginger. Mint leaves showed comparable results. However, 90% of mint leaves extracts are found to be contaminated with fungus, and repeated experimentations have shown that fungal contamination was not removed even with UV irradiation and bottling process. However, when mint was used with lime or Forsythia, the fungus in mint was totally eliminated by the other plant extracts. Results have shown that synergistic effect of Forsythia and ginger work best, and second is the synergistic effect of lime and ginger. Other results such as honey, apple cider, Coke showed commendable antibacterial effects against them both S. aureus and E. coli. The synergistic effect of Coke and ginger had also showed commendable antibacterial effects against both microorganisms. Out of so many commercial medicines that we have tested out, Woods cough syrup and Cough 100 cough syrup showed the best results. Against the control, almost all gram-negative E. coli and S. aureus are eliminated. Almost all bacteria used against Woods and Cough 100 are eliminated at a short time span of 4 hours. Why is this so? Both Woods and Cough 100 cough syrups contain a similar active compound of guaifenesin. Guaifenesin is a drug which helps to thin out and loosen the phlegm or mucus accumulated in the respiratory tract. When the phlegm is thinned out, it will be able to be coughed out of the body more easily, as phlegm contains much bacteria and other disease-carrying substances. Removing the phlegm would also remove these bacteria and disease-carrying substances. However, as all experiments are done within the petri dish and no phlegm or mucus to be thinned out and no movement of coughing to cough out the phlegm, so we concluded that guaifenesin have antibacterial effects. Apart from Western medicine, we have tested traditional Chinese medicine that are found in the market. However, results were not as strong as Western medicine, so we gathered the common ingredients of these Chinese medicine. Together with some commonly believed to cure sore throat and cough, we did experiments against the gram-negative E. coli and S. aureus. Among ginger, lime, mint, and other herbs and concoctions, Forsythia showed the best results. Research regarding ginger and mint has shown that mint actually has higher antibacterial effects than ginger. On the contrary, what we have found is that ginger have a higher antibacterial activity than mint. It has been noted that mints have fungal contamination. Priority test shows that fungal contamination is present. Even 15 minutes of UV light irradiation is unable to eliminate the fungus contamination existed on the mint. So it can be seen that fungus contained in the mint cause the antibacterial effect of the mint unable to perform. Another set of results that are on the contrary to research studies would be the results of honey. Honey has been known to have strong antibacterial activities, and microbial resistance to honey has never been reported. However, as compared to with other successful ingredients such as Forsythia and lime, it does not perform as well. Therefore, honey with Forsythia would be an ideal drink for homemade remedies, and further tests have shown that both compounds have both synergistic effects. However, on its own, the antibacterial effects are weak and shows minimal. Therefore, from research studies, the botanical source of honey, harvesting and storage methods, as well as concentration of honey used has an impact on antibacterial activity. Therefore, it could be due to the store-bought honey having little antibacterial effects. In conclusion, Forsythia, which has been used widely in many traditional herbal concoctions, showed the best antibacterial results. Studies on amount of bioactive compounds in different plants can also be further analyzed for different research studies, such as consumers can have less dependence on antibiotics. We have come to the end of the presentation. We hope you have enjoyed it. Thank you.